Um, we're going to start now. For me, it's a pleasure to introduce Alfonso. Alfonso is currently working in Barcelona, but he did his PhD in Madrid, at Universitat Computers. No, the undergraduate in Madrid, and then he did a PhD in University of Chicago, I think working on yeast. And then he moved to Developmental and Drosophila. When he came back to Europe, first as a postdoc at the LLB in Cambridge, and then he became a PI in Cambridge as well, first at the Department of Zoology, and then he moved to the Department of Genetics, where he was there for many years. And he has made very important contributions in Drosophila first. He works on compartments and boundary, and in the winglets and not signaling, where he made very, very influential contributions. And more recently, I think around 15 years ago, he started working on stem cell, mammalian stem cell, and he discovered something that we were talking today that at the beginning was a bit controversial, is that if you take a small number of mammalian stem cells, they can organize in something that is very similar to an early mouse embryo, and he called that gastruloids. And today, gastruloid is a new field. He opened this new field, and you can find people trying to study and understand the gastruloid all over the world, which is coming from Alfonso work, which I think the first paper was published in 2014, where he used for the first time the word gastruloid. Alfonso, because of all this achievement, has been recognized and received many prizes. Just to name one, the Waddington Medal, which is the most important prize given by the British Society of Developmental Biology. He has also published many books. I mean, just to mention one, that maybe he will mention here is the continuation. He's one of the editors of the Principles of Development, published initially by Lewis Wolpert. And he had made many contributions to the community. Just to name one, he started organizing some seminars which were at the interface between physics and biology, and they have been very successful. They started in Cambridge, but now they are reproduced all over the world. So, I mean, these are just some examples of the few achievements. So thank you very much for coming.
Thank you, Roberto. I think after that introduction, what you've gathered is that I'm old. There is nothing to be achieved. All you have to do is wait in life, and things happen to you. Okay, good things happen to people that wait for them. So thank you very much for coming, and thank you very much to those of you who are online for attending this seminar, in which maybe at the end I will abuse your indulgence on something. Being at UCL, and I hadn't been in the UK for 2 years, and I'm an emotional person, and it's been interesting to feel and to sense a place which I like very much and to which I'm attached, because I spent the most important moments of my life here. It's very difficult to be in UCL and not to think of Lewis Wolpert, who was a very influential. I mean, all of us that met him, and those of you who meet him through the books that he wrote, and I recommend you to read not just his Principles of Development, but he wrote a lot of very interesting books. One of them that I like is The Unnatural Nature of Science, which I suggest that many of you read. It's a little book, and it's very much Lewis at his best. So it's very difficult not to come back and think of the many discussions that we've had around here, and his personality and his influence. And of course, he coined that term that is there about how the most important event in your life is gastrulation, which sometimes you have to explain to the non-specialist. Also being here, of course, Claudio, with this very influential compilation of what gastrulation was about 20 years ago, has influenced this field, which I feel for something that is so central to biology and to our life, there is not many people actually working on it. People mention the word and zebrafish among the guides, and that's because, as you will see, it's difficult. If you're not working on xenopus or in zebrafish, it's difficult. But I think that over the last few years, thanks to stem cells, and that was the reason why we moved into stem cells, not to use them to do regenerative medicine, but to try to use them for development, and I think that that's working. That's how we run into new approaches to gastrulation, which are revealing things which I feel are surprising, and I hope to share some of them with you. So, you know, in the development of an organism these days, you get the impression that it's just about mapping genes into cell space. I mean, you definitely get that impression. And we spend a huge amount of time. I mean, this is an old way of representing single cell analysis. I mean, I don't think the reality has changed that much. This is just an old slide. You transform this into UMAPs, and the bioinformaticians will tell you 50 different ways that you will never use of how to analyze gene expression. But that's really what one gets the feeling that developmental biology is about. However, for some of us, actually, development is about mapping cells into space-time. I think there is, to me, and I think maybe this is something that Roberto was alluding to, but I'm not going to refer to a book I wrote developing this argument between the importance of look at this dialogue as two different entities that will be throughout the talk, that are cells and genes. And I would say that there is much that we don't understand about cells, and that we shouldn't surrender to this gene-centric view of the world and ask questions about what cells do that are independent of genes. So for me, as you will see through the talk, it's that development is really about mapping cells into space. And in the way I view things, genes are tools that cells use to build other cells and to build organisms. If there is a process where this activity of cells it's clearly in display is gastrulation, and particularly amniote gastrulation. That is to say, chickens and particularly mammals, as you will see. That is a mouse embryo. What you see below is some movies from Kate McDowell, who, after working through a very long time and many years, managed to film gastrulation of a mouse. And I'd like to, if you work with frogs, or if you know about frogs, even Drosophila, what you always think is that gastrulation starts when there is thousands of cells, and then at a certain moment, we don't really understand what determines that moment, but it's very precise and very reproducible. Cells initiate a huge choreography, which is very reproducible, very complex movements. Ray Keller has mapped them in the frog to an incredible detail, and it's a very complex choreography that is reproduced all the time. And I think that is a very good example of what cells can do. Of course, they are using genes for that, but that process is not being geared by genes, but is geared by cells. Things get very dramatic in the mouse, in mammals in particular. Why? Because that is the number of cells that the embryo has when it initiates gastrulation in a mouse. In the human, it's not very different. It's between 300 and 400. That's very different from a frog. That's very different from a chicken, where there are thousands of cells. Here, gastrulation starts, and I'll be happy to speculate as to why the mammalian embryo does not amplify the number of cells before engaging into gastrulation. And then, as the number of cells is being amplified, cells are acquiring fates. They're starting to move in very precise divisions, and you get an order. This is going to be very important. I don't have a pointer, but that doesn't matter. So there is an order in which the fates appear during gastrulation, and all this as the embryo is growing in size. So that is a very amazing process when you come to think of it, because you know that embryos, you have all the cells there, all you have to do is to move them around, but here cells are having to do something very clever. They're having, there you can see how the heart is formed, and there is people in the audience that know a lot about the heart. And then in the end, at the end of this process that lasts about 2 days in the mouse, you get this very clear fate map, this very clear arrangement. Out of 400 cells where there is nothing specified, and they're having, at the end of a day and a half, you get all these fates specified in order, arranged in a space, a system of coordinates. All this, to me, is an example of self-organization. And in fact, it is this system of coordinates that I'm very, very interested in, that to me is the hallmark of gastrulation. I think people say when you read certain textbooks, even the one with Lewis, I cannot change that gastrulation is about specifying the germlines. To me, the key point in gastrulation is to create a coordinate system and to arrange the primordia of the different tissues and organs in very precise positions relative to each other and with very precise sizes with regard to that coordinate system. And that's, to me, the real mystery of gastrulation. In mammals, we know that the basic system is probably the same in mouse and human. I'd like to point out to you, as you can see here, that the basic, and it's not trivial, difference between a mouse and a human is that the mouse has this cap shape that you can see here, and the human is flat. If you were to push this down, you would start to be similar, but there are other differences that I will not have time to get into. But we think that this difference probably matters for things, that there is a lot of detail that I will avoid. I just want to call this attention that the initial epiblast from which the body plan is going to be formed is slightly different geometrically. But what is important in both cases, the way you set up the axis is by interaction between extraembryonic tissues. This is a very specialized development in mammals, because the mammals develop inside. So the first thing that the zygote does is to create these cells that are not going to contribute to the embryo, but are going to provide a connection between the embryo and the mother. And that's going to be particularly the trophoectoderm. So what happens in the course of development, that interactions between the embryonic cells here in dark purple and the extraembryonic cells set up the initiation of gastrulation at one point of this sort of cap shape, which is where development starts. And here you can see how it happens. Initially, the epiblast is symmetric, is radially symmetric. There is no, now some cells, because of a set of interactions between the extraembryonic tissues, some cells at the tip, at the most distal part of the primitive endoderm, which is one, it gives rise to the yolk sac. They are specialized right here at the bottom. And then what you get, these cells are going to move. Because the epiblast is radially symmetric, it doesn't matter where they move, but where they move, they form this structure called the anterior visceral endoderm, which determines where the anterior part of the embryo will be, and at the posterior part, gastrulation will be started by the expression of brachyury, which is a universal, and I mean universal, as you will see in a minute, marker of posterior. Gastrulation then proceeds from the posterior part towards the anterior through a sequence of EMTs, which are polarized and are driven by brachyury. And here you have what I mean by being completely universal, because brachyury is a polar marker from Hydra all the way to human. So that's a very mysterious and interesting thing, but it's the first sign that there is a polar, or in general, once you get to higher metazoa, into the anteroposterior organ. And you can see here, that's the mouse. There you can see brachyury in a sheep. There you can see it in a rabbit. You can see it in a cow. It appears in all organisms. It's absolutely the same. So brachyury is the essence, the first thing that you see in the coordinate system of the animal, and is the first gene that appears in the life of the metazoa that signals this polarity. Now, the way we have been studying development for many years has been through genetic analysis. And there you have some of the very good examples. Drosophila, of course, where, as Roberto said, I like to say I did a military service in Drosophila. Drosophila is very good to get very rigorous about science. I think other thing, because you can do so much in such detail and with such rigor, that I think is very good. But of course, that screen of Nusslein, Volhard, and Bischof's opened up the study of development in other places, and it was those mutants that then, of course, there is the Ed Lewis famous four-wing fly, which I have to tell students always that that's before Photoshop, because otherwise it would be very easy to Photoshop. And that what you see at the top right is a mutant of brachyury, very early. As you can see that the embryo is shorter. It still has a polarity, but now it's lost the ability to extend, and we'll be looking at that later. So this is the way we, particularly today with these UMAPs, this is the way we understand development. I would claim that the problem with this approach is that to work backwards from the phenotype to the genotype, to what the gene does, it's not the genotype, but to work backwards from what the mutant phenotype is to what the protein encoded by the gene does is not trivial. And when you think about it, although we built a fantasy for many reasons onto this world, I think there is a lot of things that we don't understand development. And I think particularly to me, it is cells that are the link to interpret and transform the information that is in the DNA into tissues and organs. And when you think about the development of an organism, it is really, a primordium starts becoming defined and starts differentiating when it acquires a certain size. And we don't know how the genes encode that size. We know the genes respond to that size, but we don't think that it encoded. And I could give you many examples of this. I'll be happy. But so how then are we going to address a problem that is basically the activities of multicellular ensembles without using genes, or using genes in a different manner? This is in a way to explain what Roberto has said, by 15, sometimes I have to admit that it's longer than 15. In the end, I've been working with these cells longer than with Drosophila. I heard about these embryonic stem cells, which were very interesting to me, because very early on, I learned that they kept this pluripotency, this ability to differentiate into all cell types of the organism. I was very curious that they didn't make an organism, but they could make an organism if they were in a mouse, inside the mouse embryo, but they were very interesting to me. I thought maybe we could use them to learn some principles of development. Together with, I remember James Briscoe in particular, we started to use these cells to learn about development. And I think it's paying off. A lot of the stem cell biologists still don't get this, but I think developmental biologists do. Now, some people like to use this idea of engineering approaches. I like to say that we are playing with cells, really. And I'm not interested, and I'll come back to to imitate nature, but we want to learn what playing with cells teaches. I say playing because if you start working with pluripotent cells, you realize that you cannot do very much. You basically are steering the cells in what they want to do, but it's very difficult to convince them to do things that they don't want to do. And it is in this dialogue with the cells that we are having a lot of fun. As I will show you today, we are learning things from the cells, and we are also seeing ways of asking questions that would be very difficult to ask simply with embryos. So the cells that many of us have been using, and the cells that lead to this, are embryonic stem cells, which are derived from the blastocysts of mammals. I mean, these days they've been derived from most. Oh, I have to get this. Sorry. They are derived from the blastocysts of many mammals. And what is interesting, as I said, about these cells is that we can differentiate them into all the cell types of the organism, and this is being used particularly in the early 2000s. This was a chimera that will be able to use them to regenerate damaged tissues. I mean, that hasn't proven right. I think part of the reason is because really organs and tissues and embryos are three-dimensional. And although there have been some successes, and some of you might have heard from the very recent great advances of developing Parkinson cure based on the development from ES cells of dopaminergic neurons, for the most part, this idea of using these differentiation protocols to get cells that then could be used to do regenerative medicine hasn't worked out. But the reason is, as I say, because they lack that spatial organization. And the spatial organization, the 3D organization, is fundamental. And as you will see through the talk, I think sometimes, and this was very striking to me, you can have the same cells, and whether they're in 2D or 3D, they will completely different things. They are the same cells and the same genes. That says to me that there is information in the 3D organization that is not in the 2D, and that the cells are using to organize gene expression in a very different manner. And that's a very simple. So over the last few years, there has been a tendency by some people to try to create replicas of the organism. I like this difference between a replica and a model, and I hope that will become clear. Why? Because in addition to embryonic stem cells, people have been able to generate stem cells from the extraembryonic tissues that are part of the organism. And so they generated trophoblast stem cells. The trophoblast is the primordium that will generate the placenta, and cells for the Jol sac, the stem cells. So many people have spent and are spending a lot of time in trying to put together these cells. And if these are the ingredients that are making an embryo, the embryo is the cauldron of the organism, well, maybe if you put them together, they will get something. By the way, this drawing, which is very, very wonderful, is from Rosa Beddington. She had a collection of doodles that you can see, I presume, at the Creek now, and it's a very interesting collection. She was a very great biologist and also a very interesting artist. So here are some of the examples that they get. This is work of Jacob Hanna and Magdalena Zernica Getz. If you don't have a trained eye, you would think that those two things on top of those are the same. Well, one of them is the product of mixing these three kinds of cells and hoping that a miracle happens, and the other is the real thing. There are differences. These replicas that are created, they are not so much replicas, they are trying to get something that is real, but there are problems. And what is the real problem also is that the frequency with which this happens is very, very low. It's 1 in 500, 1 in 1,000. So basically, you're looking at a miracle. I mean, it's telling you something, but you're looking at a miracle in these situations. And we all know, particularly if you do genetics, that miracles happen when you're doing screens. So another possibility is to do models. And I think this is something that we've pursued. What do I mean by models? Well, you want to know what is the minimum. This is if you want a physicist approach. I've always been very interested in interacting with physicists. A physicist will do the simplest possible model and only complicate it if necessary, or if what you are doing doesn't teach you very much. So for example, you can ask what the ESLs do on their own, or you can start making pairwise combination and see if any of them produces something that has to do with the organism. And people have been playing with that. Our own version of this is work that I did with Matthias Luttel, who is an engineer, and I think this helps a lot of things working with engineers. And what you can see here is we are mixing trophoblast cells, trophoblast stem cells with embryonic stem cells, but we're not doing it willy-nilly or in a random manner. First of all, we create epithelial cysts of ES cells, and then we also create independently cysts of trophoblast stem cells, and then we bring them together. If you do it in an engineering manner, what this allows you is to vary the proportions of the cells. So you're looking now at the outcome, but varying the numbers of cells. And a thing that you will see through the talk is that the numbers of cells in these experiments matters, which of course leads to the question, what is that cells measure? Do they measure numbers? Do they measure space? What is the density? What is that they are measuring? So here you can see that we can do the experiment with matrigel, without matrigel. You can see that sometimes we start forming cysts. I mean, the cells are colored in different manners. And in the next slide, we can see an experiment that I thought was very, just to give you a flavor for this. Here, the trophoblast stem cells are labeled in green, and the embryonic stem cells, the cysts, you can see, if you are an expert and you have an expert eye, you can see that there is an epithelium there in both cases. In this case, we are starting with less embryonic stem cells than in this case, where we are starting with more. The number of trophoblast stem cells doesn't matter. And in fact, if we substitute the trophoblast stem cells for beads loaded with BMP, you will get the same result, okay? So what you will see now, the cells, the embryonic stem cells carry a reporter for brachyuria. Remember, when brachyuria appears, there is polarization. And now what you will see in the movie is that when the trophoblast stem cells touch the embryonic stem cells, you will get brachyuria on one side, only on one side. And then let's see what happens. So let's start with the small cyst of embryonic stem cells. There, you can see now brachyuria will appear in red on one side. There it is. And now you can see that it spreads, and it spreads very quickly, okay? So I thought that's there, it goes again, you can see it. Now what happens if we increase the size of the embryonic stem cell cyst? The same thing will happen, but now brachyuria will not spread throughout the whole epiblast. That's telling us that the size, the initial size of the epiblast matters. And then we can do a lot of measurements. This is about doing it reproducibly, which is what happens with these engineering approaches. And we learned that if you want to maintain an anteroposterior polarity, because that's what we are seeing there, you need a certain minimal size. So this starts being interesting because we are seeing, like in the embryo, things happen when there is a certain size of the ensemble. So then we move to the simplest possible model, and this is where the surprises arose. And it's if you have just embryonic stem cells. What can embryonic stem cells do on their own? Well, for a very long time, people had been using what they call embryoid bodies, in which they aggregated these cells to form these objects, which are called embryonic bodies, embryoid bodies that are very thousands of cells. If you read the literature carefully, what you found is that in embryoid bodies, new cell types appear that never appear when you do 2D. So that's telling you that there is some 3D organization, information that the cells are able to get. But the patterning of these cells would be disorganized, the proportions would be totally disorganized. So that's really was not the approach. But what would happen then, we get that. But then we changed the tack, and because we were interested in this, there is a lot of abracadabra here, but there is no abracadabra in the science. We spent 3-4 years learning a lot about the ASLs, and when we got this protocol to work, it didn't come out of nowhere. There is method in the madness, and I'll be happy to discuss that with you. So what we did is, when we were very careful with the initial number of cells, we controlled the chemical conditions and the aggregation conditions, we got these remarkable structures, which if you work with frogs, they might resemble what they call exogastruli, is the reverse of that. You can see in all of them that they seem to have a polarity. One side seems to be darker than the other, and the other is elongated. And we thought that, I mean, when you do an experiment, even if it gives you something weird, that weird thing resembles something about the embryo, and it happens all the time, you have to look at it. That's my. So we spent a lot of time. This is the protocol. It's very simple. We call them gastruloids. They are aggregates of defined numbers of mouse ear cells. This was very important, and I will tell you in a minute the details of that. And this is what happened. The ear cells aggregated. This is all in suspension. And after a while, this is in the center. It's a cell line that carries a marker, a reporter for brachyuria, and you will see brachyuria appearing and concentrated in one end. And then if you were to continue, that small number of cells grows after 5 days in this structure. At the top, you will see now cells coming up. That's embedded in matrigel at this point. Those cells are neural crests that are actually coming out of the anterior part of the gastruloid, and the rest is elongated. So this was extremely interesting to us. And I was worried that this was not going to tell us very much until we started to look in depth, particularly through a collaboration that we started with the Bull in Switzerland. And the first thing that we could see is, as you can see on the left, that these structures have no brain. This was very good because it solves a lot of ethical problems later. And before anybody asks me why don't they have a brain, next time somebody comes and gives you a talk about mini brains, ask them where is the rest of the body. I never understand why in mini brain talk nobody asks them where is the rest of the body, but everybody asks me where is the brain. So please avoid that question. I think I've given you the answer. But what you can see at the top is the primordium for the heart. You can see brachyury, there is nervous system in the middle. I should point out that during embryogenesis in mammals, at the end of gastrulation, the most anterior structure is the primordium of the heart. Then the brain pushes it back. But the most anterior structure is the primordium of the heart. And here it will remain so because there is no brain. But what was remarkable to me is that we could see that they had the 3 axes. They had, in the course of proliferation autonomously, they had generated the 3 axes. And they had organized gene expression with regard to those 3 axes, very much like in the embryo. These are in situ hybridizations. And over time, we saw a very similar pattern of differentiation that we could see in the embryo. In fact, for 5 days, they are following a program in the embryo. Most remarkable, and Denis Duboul was very surprised about this and has been exploiting this over the last few years. That elongation that you saw in the top right, it actually reflected the onset of Hox gene expression. Those are the 4 complexes that exist in mammals. And what we could see is over time, you can see it on the left, the opening of the Hox complexes mimic completely the opening in the embryo. And that was not only, all of you are familiar with the Hox genes, we got this pattern of Russian dolls that is characteristic of the Hox genes. So these things were during 5 days. In fact, we went to because that was important for the need to do the biochemical work that is very difficult to do with mammalian embryos. But here, we could generate this in hundreds and thousands nowadays, very reproducibly. We look at the development of the Hoxes, the onset of the Hoxes in individual gastruloids, and we could see that they were all synchronized. So all of a sudden, we have also a handle on the issue of timing, which is quite important. And during those 5 days, they are following very, very much, particularly between day 2.5 and day 5, the embryo. So this started to look like a very interesting and useful system. I've already given away one thing. If in developmental biology we use embryos and we use model systems, it's because they are reproducible. And then it allows us to do screens where we can pick up very subtle differences when we do the genetic screens, which could be very informative. So if we're going to have a system, we need for it to be reproducible. As I heard Editor Roberto say once, the embryo knows that it only has one chance to make it, so it's going to maximize it. But there is an exception always, that's the human. Only 30% of the conceptions make it to the end. That's another interesting thing that these models might be able to. So there you have frogs, maybe Lox Drosophila, C. elegans, this reproducibility, the gastruloids give us that. We can do measurements, we can do experiments. There you have an experiment we did with Pierre-François Laine. On the left is the gastruloids where we've been treated, on the right not treated. You can see that there is no gene expression. This is now, Prisca Liberali has done a huge chemical screen about polarity with thousands of gastruloids. You can do high throughput. So that's why it's becoming a very important thing. But the important thing, and this is going to be the second part of the talk, is understand how this relates to the embryo. You will have noticed that they don't look like embryos, although they have, and I hope you realize that the gene expression is very organized, very much like the embryo. So we need, in order to use them, we need to understand what they are. The first thing that I mentioned is that and this starting to give us insights because of this experimental robustness of the system, we can ask the question, does it matter, the initial number of cells? And if it does, I mean, why and how? So here you can see this is part of a preprint that we put, I mean, the paper is under review. We start with different numbers of cells, we treat them in the same manner, and we can see what happens later. And what happens is that if we have about 20 cells initially, gastruloids formed by gastruloid, I mean this elongated structure. Sometimes they form, sometimes they don't form. If we have too many, we start getting Siamese gastruloids. Within a certain range, we get very, very good gastruloids, okay? So that's telling us that the initial number of cells matter for this structure. And we'd like to know why and how, and we are in the process of doing that. In the meantime, we are learning things. For example, this elongation, initially you get a cyst of cells that then gets a polarity and it starts elongating. And what we could see is that smaller gastruloids lead to an earlier elongation. I mean, that's a summary of the data here, but we can see that. Why is that? I mean, we are trying to think that the cells are more pliable in the smaller gastruloids than they are in the larger ones. And that could be a reason. And this preprint now has a lot of experiments showing that something about the rigidity, something about the biophysical properties of the system matter for the implementation of these signals that lead to the elongation. We also notice something equally interesting, that the smaller gastruloids tend to favor neural fates, and that you only get mesodermal fates as the number of cells increases, and then you get a mixture of both. And you can see that in this slide. I mean, that's what happens. And then you can see that here. So this, yeah, this is one of these repeats. The smaller ones have to do with, they get mostly neural, that's SOX2, and then you get a combination of neural and mesodermal as you increase. We think that this has to do with the ability of cells to interact with each other and nodal signaling at the paper, and that's why the recent very fascinating paper from Nicoletta Petri about the biophysics of cell ensembles regulates nodal signaling. We think that it could be the same in this case, and we are thinking of so the way we know the brachyury pole appears, it doesn't appear magically. We know that cell sorting is very important, and that depending on the size, what we think is happening is this: there appear different foci of brachyury in the aggregate. Then something, I think that brachyury is not the signal that determines the polarity. I believe. Believing means, as a Catholic, to believe things that you haven't seen. So I believe that the signal is probably mechanical, and then you get an aggregation of the brachyury sort of patches into one. If the epiblast is very large, you might not get all to converge in one place, and you might get two. In fact, I would surmise that Siamese twins are related to a very large epiblast, larger than normal. We are looking into this in great detail. This is just to give you a flavor of the kinds of things that now we can ask that relate to the embryos. Why morphogenesis starts at a particular time and a particular moment when there is a certain size? Well, I think we have now a system to answer that question. This is a fascinating observation from Thomas Gregor in Paris, which is that within that range of initial cells where gastruloids form efficiently, they scale. And scaling is a very deep problem in biology. And I think, again, we have an experimental system that is going to allow us to do that, all based on the reproducibility. And the reason why we call them gastruloids is because these structures at the end of the feet, they represent very well an outline of the body plan at the end of gastrulation. One of the things that we did, instead of doing one gene at a time, we teamed up with Alexander van Oudenaarden in the Utrecht, the Hubrecht Institute in Utrecht. He had developed this technique, which is modern histology, in which you take an embryo. He had done it with zebrafish, we did it with gastruloids, and you section it very finely. I mean, what you have here on the right is expansion microscopy that allows us to get more sections and therefore better resolution. And then with every section, you do RNA sequencing, and then you can get a spatial organization of the genome in space. And you can see here, this is 5 gastruloids taken at random, and basically they are highly reproducible. Not only, I mean, I was very surprised that the genome was reacting. We compared this to embryos, and the genome is reacting in these structures very much as it does in the embryo. But I want to remind you, it doesn't look like an embryo, but it has. I find this is still very remarkable, that the proportions of the different primordia as reflected in the genes that determine those primordia is exactly the same size and the same relative size as in the embryo. So the genes are able to measure a space, or space is able to using the genes in a manner that is independent of morphogenesis. This is very clear. This became very clear when we came to notice a very strong signature of the process of somatogenesis, which, as you know, is the generation of the somites that will generate the thoracic cage and the muscles of the main body. You can see here, this is from the gastruloids. You can see that we have genes that are associated with the presomitic mesoderm, genes that are associated with the somatic mesoderm, and even a gene, RIPLY2, which determines the boundary between the presomitic and the somatic mesoderm. This is a gastruloid which, although it's expressing in the right anterior part genes associated with somites, there is no trace of somites. So I will show you in a minute. We can compare this to the mouse. This is a data set that Olivier Pourquier produced a few years ago, and we can see that this is exactly the pattern of somatogenesis of the mouse with the same proportions. And as I said, here you have, this is a chicken actually. You can see this boundary here between the presomitic mesoderm and the somatic mesoderm. This is a gastruloid, no trace of the somites. Now, I want to pause here because I'm still very surprised that the genes are able to measure a space, if you want to use a gene-centric view, in such a precise manner without any morphology. I mean, this is food for thought. Now, in the field of organoids, which is more alchemy than a science for now, although that I hope is changing, there are two components which I like people to think of like Tinkerbell powder. You remember Peter Pan? Tinkerbell spreads some powder over you and then you fly. And I think there is Tinkerbell powder in the organoid field, and the components is matrigel and wind. Matrigel, nobody knows what it is, but just sprinkle matrigel and magic happens. So we decided to see if we could play Tinkerbell with the gastruloids. And when we play Tinkerbell with the gastruloids, I mean, they are in suspension, but we start embedding them in matrigel, we start getting these structures, which are somites. In fact, this is a gene that is expressed in the somatic region, and when we put it in matrigel, it starts segmenting. As we got the technique improved, we start getting these remarkable structures. So those genes would be mixed in the normal thing, but now we start getting this remarkable segmentation, which is a prelude of somatogenesis of the somites. And in fact, Jesse Vibliet, now in Dresden, managed to get, playing with the conditions of the matrigel and the signal, these remarkable structures which now have spinal cord, and they also have somites, real somites. So what this is telling us, what this is telling me, I mean, also we managed to make gastruloids from human, which in the end is the very important thing. I'll come back to this in a minute, but again, we have reproducible. We, in fact, were able to see very much the same thing, germ layers, the same kind of organization. In fact, we were able to, through a certain reasoning, we saw a signature of somatogenesis, so we knew that in these gastruloids, they had completed the phase of gastrulation and they were starting somatogenesis. That was, we wondered if it would be the same as we saw in the mouse. There you have somatogenesis. We can see the clock working. And again, you know, transcriptional FAMAT and oscillatory dynamics that not require matrigel, but the formation of the somites requires matrigel. So we get again a situation in the human in which we can get this genetic map of the organism without a morphological map for the most part. Now, to me, what that said, thinking about this, is that there are two components when it comes to development, you know. One is what we think about all the time, which are gene regulatory networks, which depend on signals and genes, and this is what we play around. Now, I think these and other elements are talking to me that there are something I would like to call cell regulatory networks, which have to do with mechanics and geometry. In fact, we did publish a commentary in the opening issue of the journal that Roberto edits, in which we put forward this idea. And I think I'm interested in exploring if we can build cell regulatory networks in the same way that we build gene regulatory networks, using cell-cell and cell matrix indentation, traffic, polarity division, as a totally different and separate entity. I think right now we think to represent this, I don't think these are represented in any space, but I would say that development depends on the interactions between gene regulatory networks and cell regulatory networks. In fact, this is from a discussion many years ago, in which I think Pavel Tomanchak put forward this idea in a very nice manner. I find strangely liberating to think of genomes and emergent cell behaviors. That's what we are talking about, as two rather independent entities that need to remain compatible with each other in order to create a functional living system. In fact, I would say that these two systems, the interplay between these two systems, which are being revealed when you think deeply about these organoids, the interplay between them is at the root of the variability of morphology that exists in the animal and the plant world. But now I'd like to come to what is the central element of the talk, which is gastrulation. And just to remind you, and these are very important things, gastrulation in the mouse is seen as dependent on the interaction between wind and nodal signal. And when you read the textbooks and the reviews, they will tell you that this is an additive or a synergistic interaction between wind and nodal. That's what drives the appearance of the primitive streak and the movement of the primitive streak from the posterior distal part of the embryo to the anterior. Don't worry about the cylinder, it's a very strange geometry, but if you turn what is basically a calzone into a pizza, you have exactly the same as a human embryo. What we know, as I say, is that during time, there is a series, a sort of very nice time series of assignation of cell plates. Initially, the endoderm, there is what is called the anterior primitive streak, which is going to generate pretty much everything up to here. Then there is a switch at the end of gastrulation. Gastrulation, as defined by Claudio, ends up when the node appears at the most distal part. And now you switch to a second program, which is going to get the axial elongation, which is what the formation of the caudal lateral epiphysis. The brain is specified independently, and I think the gastruloids are telling us, by an interaction of elements of the node with the anterior ectoderm. I'm not going to get too deeply into that today, because I'd like to highlight these two modules during gastrulation, which is the anterior primitive streak, which specifies everything up to here, the level of the forelimb, and then the rest of the body, which is specified by different systems. Now, what we knew, and this is something the work of Sebastian Arnold in Freiburg has shown, that these two sort of modules are under transcriptional control by two T-box transcription factors. One is mesodermin, which controls all the defects in the anterior primitive streak, and the other is brachyury, which controls all the posterior one. Okay, and he has shown that in a very, very nice manner, and that what you get during gastrulation is a progression from this mesodermin-dependent program to the brachyury one. Those of you that know about mouse development will know that both mesodermin and brachyury are co-expressed in the primitive streak. But what Sebastian has shown is that mesodermin is suppressing the targets of brachyury, and only when mesodermin disappears at the end of gastrulation, now you get brachyury to work. So there is a logic here, and that is very. Now, something that had always bothered me with the gastruloids that we've been selling and that most people use is something that the Denis Bull always teased me about. He loves it because he can study this axial elongation and the hoxes, but he always pointed out that in these gastruloids, the anterior, the representatives of the anterior primitive streak are very sketchy. These things hardly have endoderm, they have in a very variable manner some cardiac progenitors, but it is true that they mostly represent the second program that is wind dependent. That maybe is not surprising because they depend on chiron, chiron is wind, we are activating mostly the second program. Interesting that they jump, but I will explain that to you in a minute. So there you are, what we have here is an early anterior program that is very stochastic, and then a very robust posterior program. When we look at what happens during the pulse of chiron, which also we put chiron because that's the way you initiate gastrulation with a little bit of wind, what we notice is that what the gastruloids do is what the embryo does. It starts through the anterior program. So there seems to be a deeply ingrained program of gastrulation that is going to start with this phase. This, by the way, is a very ancestral program because it happens in all organisms. And when we look at the genes, what we can see is that initially, in a very stochastic manner, this is the gastruloids are starting to express these anterior primitive streak genes. However, and that's indicated here, what happens is now the chiron comes and completely shuts off this program and switches the system into the posterior one. This is something that is very surprising, that you can jump stages, that you can jump in the development of this, particularly the embryo stem cell models, that you can bypass stages and continue development of very posterior things without having gone through the normal development. But that's what happens. And then, as I say, what we get mostly is the caudal lateral epiphysis and axial extension, and we completely obliterate this anterior program of gene expression. We can see that also when we look at genes, and you can see, sorry, You can see that here. I just want to show you that we are activating in the thing all the Hox genes, which are what gives you the. But we don't have any of the anterior genes. This is normal. Now, because the gastrulation starts with nodal, we wonder what would happen if instead of chiron now we substituted it for activin, which is a proxy of nodal. And now what we are seeing here is that now we are doing the reverse of what we're doing with the chiron gastruloids, and now we are activating and getting the anterior program with very little trace of the posterior program. And you can see that here, now by maintaining nodal, we are maintaining all the anterior program and we are getting very little of the posterior program. In the gastruloids, what this is telling us that these two programs are totally separate. In the embryo, they might be continuous, but here we can activate, we can send the cells one or the other. And I find that very, very intriguing. And you can see that in the one. There is a little bit of wind. Nodal does not repress wind continue, and we know that wind probably is important for some of the phase that we need. So what we have, therefore, we can build gastruloids that are anterior or gastruloids that are posterior. And you can see that as if these modules are totally independent from each other. And in the embryo, somehow, they are put together. You can see that here. So we think that when we measure them and we look, we play with conditions, what we think happens here in gastrulation is that the levels of nodal decrease over time as the levels of wind signaling increase. And that is what controls the transition from the anterior primitive streak to the posterior primitive streak. We also know that nodal and wind are, the way that the system works is that wind is down-regulating nodal. And that we have shown in the other manner. And it is as wind increases, you turn off nodal, and therefore you are starting to activate the program. One thing that comes from the work of Sebastian Arnold is that iomesodermin and TBXT are not only involved in specifying fates, but they are involved in getting the cells out of pluripotency. If you make a double mutant iomesodermin TBXT, the cells don't leave pluripotency. And I want to remind you that particularly brachyury is a very ancient, very, very ancient element in the development of organisms. So we have this program in which, as I told you, first both of them switch up pluripotency. They throw the system into gastrulation, first driven by iomesodermin. Then when nodal goes down, you get activation of the targets of brachyury, and you get the second part of the organism. Now, something that we realized when we were looking at wind is that I told you that when we put chiron is a proxy for wind signaling. What we notice is that you were skipping stages, that you were jumping from anterior to posterior. But what we notice is that depending on the concentration of wind, the jump would be lesser or longer. So it looked to us as if wind was not regulating fates. We couldn't see a change in the amount of mesoderm or in the amount of endoderm by changing the amount of chiron that is of wind signaling. But we did see changes in the stages that were represented. The more wind, the more posterior, the later. So that led us to suggest that perhaps, and we can see this in a variety of manners, just to show you that we are doing our homework. Here you can see particularly the hoxes are very obvious. As we increase the concentration of wind, we get more and more hoxes, and we get the suppression of the anterior program, all in a very graded manner. So wind does not seem to be regulating fates, but regulating the timing of events. In fact, we think when we do the same experiment with nodal does regulate fates. So we think that what we have is a system in which the concentration of nodal is regulating the fates, and wind is regulating the concentration of nodal by negatively regulating it, and at the same time regulating the timing of development. And I actually think Roberto mentioned my interest in Drosophila, and I spent a huge amount of time, and to be honest, that was the reason why I moved to these cells, because I always felt that wind is not a signal in the sense that we think of signals of controlling fates in this abstract manner that we think, but wind was coordinating events in time and in space. I mean, I'd be happy to elaborate on that. So it's very good. We have a lot more experiments that at least in this system is very clear that wind is determining the timing and the coordination of the events within the. Now we have a system now in which we can generate at will the cells. We let them go through gastrulation, and at the end of the 5th day, we can start moving them into different systems and make endoderm or make cardiac primordia. make spinal cord. We are even working on the blood in some collaboration. So it's a very versatile system, which is teaching us, I mean, what is that we are learning with the gastrule is that the embryo is a modular system. This is a very surprising thing to us. I mean, not only just in terms of the primordia of the organ that can develop them independently, organoids would have told you that, but I've shown you that we are starting to take apart modules that in the embryo, it would be very different, very difficult to sort out with genetics, that is nodally or mesodermin wind brachyury. Show that ES cells have a potential to self-organize a coordinate system, that the extraembryonic tissues bias this potential. You might say in the embryo, I've told you that genetics tells us that the extraembryonic tissues are setting up the coordinate system, but it was implicit. I forgot to tell you that the gastruloids do not have extraembryonic tissues. So what that's telling us is that the ES cells themselves have the ability to generate a coordinate system. Now, in the embryo, what happens is that you don't want to have an apipolarity in every which way in the embryo. And I think what the extraembryonic tissues are doing is sort of biasing that axis into particular ways, because we know that the order of the cells matters for the interaction with the mother. And we have experiments to that. That cell-phase assignments are independent of morphogenesis. This is a very surprising thing, to which I will come back in a minute. And that, to me, these programs of gene expression, the gene regulatory networks, control time and programs, whereas morphogenesis controls space and patterns in space. And finally, and this is more for the stem cell community, the importance of the starting conditions. As I always say, we start from one cell, and the embryo takes a lot of care of that cell for a reason, because there are many things that we cannot reproduce from these embryonic stem cells, which probably are telling us about the conditions. I think what is important in this field sometimes is to highlight what the differences are. Many people lead you to think of the similarities, but I think it's in the differences that we learn and that make us think about the experiments. Now, we did human gastruloids. I don't want that. One of the things with the human gastruloids is we have very good references for the mouse gastruloids, because there is a lot of work done with mouse embryos and with, but with humans we need to see whether the gastruloids that we are making, how particularly because they have these very abstract forms, we need to know how they relate to the embryos. And it was very good a few years ago. This is very difficult because it's very difficult to obtain human embryos, and of course to do the kind of descriptions or observations or experiments, if you wish, that you can do with animal embryos. And we have to rely on chance and on opportunities. A few years ago, Shankar Srinivas came across one embryo during gastrulation. They did the obvious thing, which was to do single-cell RNA-seq, and they compared the single-cell RNA-seq of this human gastrula with our gastruloids that position those cell fates in space. They could find them all, and I think this is so far the validation that we have. There are others coming. I'm engaged in a collaboration with some people to try to create a library of very early human embryos that will give us insights not only into the early human embryos, but also to benchmark these models that are becoming now very popular. In the meantime, also, another thing that we can do is try to build gastruloids from animals closer to humans, but that you can have an easier access to embryos. And of course, there are monkeys, and I'm sure there is people doing that, but in work that we started a few years ago with Ramiro Alberio in the University of Nottingham, they've been working with pigs. They've been doing pig gastruloids. They've also been doing cow and sheep, and that's quite interesting because we can also see now start comparing both the embryos of the pig to the gastruloids of the pig, and now the human gastruloids with the others, and start triangulating. And it's just to tell you that these comparisons are going to be useful and are going to be help us benchmark and also learn the differences between species that might have importance practically. But what it's, I'd like to finish, if you give me a few minutes to indulge on a topic that is very fascinating to me. So I've told you that gastruloids, haven't said it, but let me say that many people that know about amniote gastrulation will tell me, well, there is no primitive streak. What I have told you, without being explicit about it, is that the gastruloids are the primitive streak. And what we are showing is that the primitive streak has a self-organizing capacity. And it comes into modules where wind is regulating time and nodal is regulating space. Now, this is a very amniote thing. I mean, there is the primitive streak, And you know, we know, and I've mentioned that in evolution, organisms inherit programs of gene expression. So mitogenesis, cardiac, I mean, they build on those programs. But we wonder a few years ago whether in evolution, if we go away from the amniotes, whether this ability of self-organize existed in an amniotes, in fish. And working with Ben Steventon, at the time I was in Cambridge, we decided to see if the pluripotent cells of the zebrafish were able of doing the same as the mouse pluripotent cells. And the result was very remarkable. This is one of the very early experiments in which you can see this is brachyury, and it's just remarkable. I mean, when they showed me this, I couldn't tell which was the fish and which was the mouse. And in the end, you get these structures that are extremely similar to the ones of the mouse. This is the fish, and even on one side it has this structure, which is the cardiac structure, which in both cases is pumping with the same frequency. This was very, very, very remarkable. Ben's group went on to show, because of course there is a lot known about the zebrafish, and they had to prove that there was no pre-pattern. And many of the things that they show in this remarkable paper to me is that we saw very much the same things that we had seen with the mouse gastrulation. You needed a certain size was minimal, and really, at least to my satisfaction, many people still might argue that there is a pre-pattern. They jumble the cells, as you can see here, they remix the cells. So this is revealing that this ability of self-organize that TS cells is a very ancestral capacity that cells have, and that we need to understand. Now, gastruloids have been made. In fact, Jeremy Green, inadvertently many years ago, did something that now he realizes that it was a gastruloid. You have here zebrafish, this is mouse, this is pig, this is human. I could tell you now many more. Somebody made, just for folkloric reasons, gastruloids from rhinoceros. Don't ask me why, but that's, I suppose people get very excited about these things. I'd like whales, but there's no whales. Now, I want you to notice one thing. The embryos are very different. The embryos are very different, but the gastruloids are the same. This is very surprising, because you would expect that the geometries of the initial, the pre-gastrulation embryos is different. And you know, the whole process of gastrulation is different. So how come we are getting the same structure? To me, I think this goes back into this idea that there is the gene regulatory networks and the cell regulatory networks. And I think what we are seeing here, I'm going to elaborate in the next 2 or 3 slides, is what the cells can do when you eliminate their boundary conditions. That what generates the differences between the different embryos is not intrinsic. This may be a very ancestral form of animal development. And what you are doing is creating variations of this by changing the organization of the egg, the boundary conditions of the embryo, and imposing these cell regulatory networks on the gene regulatory networks, it was create the differences. But at the heart of that, there is what the gene regulatory networks do. So gastruloids reveal a morphogenetic ground plan, a platform for the integration of those 2 networks in the building and the dosification of animals. Now, it's interesting to me that Turing, in his very, very famous and great paper, amidst other things, he does make a point in the introduction that a biological system is a complicated interaction of chemical and mechanical inputs. And he makes the point in his paper that if you want to understand and to work with that, being the genius that he was, you have to separate them. And in his great paper, he goes, I'm sure intuitively for the easy one, that is the chemical. Because he does say in that introduction that the chemical component adds an incredible complexity to the chemical one if you want to work with it. All that is in the introduction of his paper. And I think what the gastruloids are showing is that he was very prescient, and that indeed these systems have 2 totally different components that somehow come together in the generation of an organism, a chemical and a mechanical. And I would say that this is one of the very important contributions of this system of ours. And that brings me back to this idea that has been around for a while and that has been put forward very clearly by Denis Debout over the years, of the hourglass that some of you will have heard of, in which it is noticeable that there is this state, which is called the phylotypic stage, in which all embryos, independently of how they start, they look very, very similar to each other. Although they start from big differences and they end up in big differences. And for many years, he's been wondering, what are the constraints? What are why are we going through this neck of an hourglass? In fact, we know that you can transform one embryo into another by changing the boundary conditions. This is from a very nice review of Rosa Beddington and Liz Robertson many years ago, which I like to use in Louis Wolpert's book. And so what I think this is saying is that the differences here, the differences at the bottom of the hourglass, I should also point out, and I've discussed with Denis, most people paint the hourglass the other way around, with gravity driving the development of embryos. Then he would say that you need energy. It's an elevation from the embryos towards something higher. So that's not a mistake in the way he's drawing this hourglass. But I would say that indeed what we are doing with the gastruloids is eliminating all those initial constraints, boundary conditions, and we are revealing the phylotypic stage before the embryo does it. Because gastrulation to me is not only about creating the coordinate system, providing the localization of the primordial, it's also about removing all these boundary conditions and all these constraints that are generated by the reproductive biology of the different systems. Ben and I have written a little bit about that. So the gastruloids are revealing something very, very ancestral. This thing here is not a gastruloid, it's an ascidian embryo, which I think it's revealing this very basic, ancestral, fundamental form that is revealed to us by the gastruloids. So with many thanks for your attention, I just want to also thank the group that has assembled in Barcelona. I really, it's been great fun I think we have now a good understanding of what gastruloids are that allows us to use it. I find interesting that physicists use it as a way to understand symmetry breaking, mechanical constraints, because it's a system that represents many features of the real embryos without being real embryos. And that's what interests them. For some of us that are more of a developmental bias, we are starting to see development in this modular manner and to start asking questions about the interaction between these modules that you cannot ask in the embryo because the embryo is an integrated whole. As I said, a lot of this has happened in Barcelona. When I left the pandemic, I think I had lots of questions about gastruloids, and I wasn't sure that they would be useful, but I am glad that they can be a good model for studying developmental biology of mammals and maybe of other systems. And of course, the funding bodies, particularly being in Europe, and I'm very glad that the UK managed to get into the ERC. Some of the human somatogenesis was funded by the MRC. And as I say, thank you very much. Look forward to your questions and to talking to some of you later. Question?
Yeah. What is matrigel doing? No.
No. I don't laugh. I don't laugh. I say.
It's not that if you put that matrigel, they put like morphogenesis and form somites.
Okay.
So it's just a matter of physical constraint, or it's about signals contained.
I think you could ask the same any organoid, particularly adult stem cell organoid, the old use matrigel. I now know that there is people. So matrigel, it's people have studied matrigel. There is over 1,500 different protein compounds in matrigel. It's very, very difficult. People for many years were trying different gels that do not contain physical, and they never have got something with the same properties as matrigel. Now, over the last 2 or 3 years, a few labs are starting to get something that might be able to separate these two very important components. I mean, cells, don't have to say this in front to Roberto, they are very, very sensitive to the biophysics of their environment. And I think matrigel is a very crude way in which whatever mechanical is tempered by the chemical environment. I think that's the minute we understand matrigel, it's going to be very, very useful. But it's very difficult. I mean, Matthias Luttel, my friend and colleague, spends a lot of time thinking about this. And as I say, I know that things are coming that are able to tease these two things apart. And that's going to be very important because I agree. So in fact, in those experiments where we put the gastruloids in matrigel and we see disappearing, we don't change very much the transcriptional profile. So it has to do with the translational, which is something that we forget in this UMAP world in which we live. Or also, a response, it has to be mostly a post-transcriptional response to whether it's, we know that, probably integrins, but we don't know. It's telling us about the world.
Yeah, so with as controlling timing, is that something that is like a bespoke way of interpreting things in the gastrulation case? Or are you saying wind is controlling timing in, I don't know, digit patterning or like, you sort of hinted that you're thinking this is cool.
So this is not something that just comes out of this. In fact, this was the reason why I moved to ESL, because I wanted the system so I get a hint of this in Drosophila. I could tell you I've written a lot, but it's very boring. It's very interesting. It will happen to many of you. You do something really interesting, but it requires a lot of words to explain, and people don't pay attention to it. You discover something that people can use to write papers, and then you'll be given credit to. Okay, so but to me, the wind story is a very important one, because when you look around, and as I said, I've written quite a bit about this. Wind appears everywhere. Wind is part of Tinkerbell, and people don't want to think about what it does. When you look carefully, and we looked at this very carefully in Drosophila, and we have data in gastruloids also, the phenotype of wind, when you look close to what happens when its cells become very uncoordinated, extremely uncoordinated in the patterns of gene expression, and in the what happens is that wind is always necessary. I could give you, I don't want to give now talk on wind. I'm just going to tell you that this statement doesn't come out of here. In fact, it was good that it fits. So the next step is also going to need wind, but you know, start with cells that are uncoordinated. So now you're going to get stronger phenotype because wind is necessary for all the steps. Sometimes people wait to see the phenotype of wind. What we see when you put a lot of wind is that things happen much quicker. That coordination we've written a lot about this. So we have, there was a basis to think about this. If we don't put wind, the gastruloids are they develop polarity, some do, others don't. There is a lot of heterogeneity. When we put the chiron, everything becomes synchronized. Wound healing, for example, in wound healing, particularly in regeneration in the zebrafish, the first thing that gets activated is wind. And this is very well known in the field. There is even a promoter in the Wind1 gene that is specific to response to wound. And I think this is a way of the system to make sure that what it's going to do now is very efficient. So to me, wind, call it coordination. I think spatiotemporal coordination. Cells in large cellular ensemble, cells are making decisions on an individual basis. And we now know that there is a lot of stochasticity. Let's use that word for that. Wind is removing that variability. And when you start looking at the literature, this is really what it's doing. It's making the system robust. And that's what we are seeing here. Okay, the timing, because it's been tempered by different concentrations, we can time the thing, and we are doing a lot of experiments with that. By timing, I mean, it's this coordination of events that is so important. And in Drosophila, it was very clear. The more wind we put, the faster the things happen. A very good example was, which really struck me, there is a set of progenitors, if people from Drosophila will know the muscle progenitors in the embryo, they appear in very precise pattern relative to stripes of winglets. You remove winglets, and there is nothing. There is no pattern. Now you express activated winglet signal everywhere, the pattern appears in the right places, but faster. So again, nothing to do with the pattern. And Massa, of course, remembers from his Wind11 experiments, where you can, in a lot of the phenotypes of the segment planar cell polarity phenotype, you express wind everywhere, and you get normal pattern. It's only the dose that you have to temper. But wind, to me, it's a coordinator of events. And I could bore you to tears with this, but that's your tears, not mine. I mean, I would be very happy.
Yeah, Mas.
So in your model, the gastruloid, the first step would be more posterior.
The traditional ones. Now we have the anterior ones.
Yeah. Then by adding or activating nodal signal, you would get more anterior or mesodermal structure.
Well, no, mesodermal, anterior mesoderm, and endoderm.
Yeah,
very
and
important.
then my question is, can you get a functional heart beating?
No, but in a small proportion of them, they do, and work particularly. I don't want to talk about other people's work, but now people are exploiting this. What it's interesting is that now we can do Lego. So we can get the anterior and the posterior together, and amazing things happen. You get pharyngeal arches, for example. So the things now start interacting in a manner that are able to start reproducing. Now we are also interested in putting the brain there. So we are a piece of Lego. Cellular Lego, in which the genes are just the colors, the shapes of the Lego. As Louis Wolpert, and I've seen him do that in a big meeting with hundreds of people, repeat after me, genes are boring.
I have a question related to that. I mean,
To the genes are boring, we can talk a lot about it.
yeah, no, I mean, there are many things of this gastruloid that are very surprising to me because I have the idea, probably wrong in development, that you really require every step of development
I know.
to progress to the next one and the next and the next one. So this thing that, for example, you have modularity, where you can jump from one step, is very surprising. So my question related to that, and it's coming a bit of the question that people working on cell migration, is that for many years people have been working on cell migration in vitro, and the cells can do amazing things. But when you test them in vivo, for example, the embryo, the repertoire is much more limited. So my question is there any evidence that some of the things that you have found in the gastruloids, like for example the modularity, really happens happening in the
No, this modularity, I told you, nodally mesodermin, I mean, that's, and we have two preprints now with Sebastian Manor, one very focused on the transcriptional networks, the other in the signal. I think you've said something, I think those of my generation have been brought up to think about development very intuitively, that there is the whole, that there is not into pieces. And I think a lot of people also, they just write the papers, don't think. Thinking is very important. And I think today, sorry to say, but I think you have to combine both things. And I think these embryo models are telling us something that is very surprising, because it's not just the gastruloids which are able to jump over the blastocysts and the epiblasts and generate these structures. Blastoids or epiblasts, a lot of these models are jumping. They're accessing these states without having gone through the other ones. And I think we need to pause and think about that. I mean, I'm going to be a bit perhaps exaggerated, but I laugh, as Roberto has said, I have lots of friends that are physicists, and they used to say quantum mechanics is very strange. If you think about this whole world, it's telling you very strange things. I think what we are seeing is the elements with which evolution works. And to make an organism requires the integration of these pieces into an organism. That's the role of evolution. This is telling us the bits and pieces with which evolution works. Evolution has managed to create, I like to use the term entangled in the way a physicist would, these things that they look causally corrected, but all they do is fitting because the organism needs to reproduce. But we are seeing in these models. I mean, I'm very surprised that people don't stop and think that you can make a liver out of nothing without having gone through an embryo or something that resembles a liver. So I don't have an answer to your question, but I'd like to people to point to notice what you think. We see the elements, and sometimes we need to carve a little bit in the system to see these elements, but they are there. To me, the gastruloids are making me think, look at the embryo in a very different manner. I'm sorry I don't have the answer, but I want to emphasize the point that you have made.
I think it was
Octavian.
I have a very silly question, a very naive question.
Which gene?
I'm brains. Why?
Which one?
Where's the brains? Where does the size come into play? Because gastruloids are quite different in size.
No.
But
That's
the
the point.
are, so
Oh, no, the gastruloids are all pretty much the same size, within 10%.
Okay.
I mean, this is also telling us some control. This is a very good, and the scaling that Thomas Gregor has shown in a very striking manner, because it's very measured. I mean, this is going to allow us to explore all these things.
I'm sorry, and those numbers of cells or the size that you have had, how are compared with the embryo?
Pretty much the same. It's about the epiblast. When it's going to start gastrulation, it's about 300. In fact, a colleague of mine in the lab, there is a very striking phenomenon in mammalian development, which is cell competition. The embryo, just before gastrulation, gets rid of the cells that are not fit, and he's been able to reproduce all this in the gastrulate. There is a whole preprint of it, and therefore, again, it's going to allow us. I think the reason for that is that the embryo is going to engage in gastrulation, which is, in mammals, it's a very heavy, all this proliferation, all this, so you don't want to use cells. In fact, if somebody rescues those cells that are unfit, Miguel Torres did, you get tumors. So the system is
Right. Start at the start, I think at the end. The only thing they need to but at the start, you put yourself together and they'll obviously migrate towards one another as a co-attractor.
No, it's a round bottom plate. It's gravity.
Okay. And then at the end, I should have known from you that it would be physical, not chemical. At the end, it seemed like the neural crest cells were coming out normal gastrulate.
Well, yes, there is matrigel there. At that stage, they are walking into the matrigel, which is what they should.
The matrigels everywhere. But
Yeah. Well, because the film that you're looking at the plane. So I haven't given you the 3D. It's a focal plane of one plane.
Still coming out normal. I've granted you a start, but at the end, there's got to be some information there.
Oh no, I'm not denying that there is. Yeah, I agree. Even if surrounding, they are going to be. Yeah, they're not going. Yeah. I don't know in the more. I mean, I hope the people I'm talking here, and I hope the Zoom people are, because I hear better there. I think people now that are building systems where the nervous system and the somites, and they get neural crest at the top, I think it'll be interesting to. I mean, there is people doing that now at secret points. Unfortunately, I don't want to interfere, fortunately, with Roberto, so I don't think much about the neural crest. Robert, do you have another question?
No,
Okay. Yes, please.
Yeah, so I was thinking about the point that Roberto raised, which is the decomposability or modularity that exists. To think about like the things that respond to wind, for instance, in the embryo.
The what?
Things that respond to wind. For example, we have those called chordate.
Yeah.
That is like the antagonist that
Yeah.
sets up the axis. And the classical studies have shown that these genes have response elements that respond both to and activate or normal signaling. So I'm just wondering, like, you know, I mean, obviously, like having an excess of wind will also activate them, but in the actual embryo, like both of them have to work together in a certain.
As I said, so what we know is that the guscoid, all those genes are in the anterior endoderm, which is going to give rise to the precordial plate, which is an element of the node in the. And we can see that we see very little of that in the posterior gastrulates, but they appear very, very well in the anterior one. So again, this is helping us see that. So we see all those genes associated with the anterior mesendoderm and the precordial plate only in the anterior ones. We are now seeing what happens when we put them together. We can see them migrating to the right place and doing. But so I think I like these abstractions that these cells are doing of the embryo, because it's teaching us, leading us to ask questions in the embryo that we wouldn't have thought. I think this is really bringing us to the self rather than to map genes in a manner that it's all very predictable, and there is no discovery when we know that there is a lot. And we can ask questions now about those things. And again, the proportions are very good when you start with which is the thing that strikes me, you know. The primordium will start growing when it reaches a certain size. This is something that we obviate, but this is true. Why? What is that? So here we have a very simple thing. The program will start when there is a minimum size. Now we can explore this. Not going to be easy, but you know, there's a lot of young people here and they have more imagination than I do.
Okay, so with that great sense, we can finish something.
Vamos a ver. I think